All right, so it's my pleasure to be here. Thank you again very much. And I will be telling you about work from our lab on making fluorescent and bioluminescent proteins for reporting biology. And since this is also a course, I'll try to make it understandable and provide a little bit of background. But I think what's interesting about making indicators is that we get a lot of inspiration from Marcel Proust, from À la recherche de temps perdu. He wrote, the only true voyage of discovery would be not to visit strange lands, but to possess new eyes. And in some sense, the quest for biological indicators is like trying to build eyes for biology. So I think we all use GFP. It was revolutionary because it was, in some sense, a new eye for biology. It was discovered by Osamu Shimomura in jellyfish. And then it was worked on by Roger Chen, improved, and used to build probes. We've already seen movies of Fuji. Fuji is just such a nice example of how even just a very basic act of fusing a fluorescent protein to another protein allows you to see some aspect of biology that would otherwise be hidden. In this case, green marks cells in S phase and mitosis, and red marks cells in G1. So I'll tell you three aspects of molecular probe development. First is our work on voltage indicators as an example of a very fast kind of indicator. And then it has implications for how one does microscopy as well. Second will be doing something completely different, looking at bioluminescence, and we could look at some of the advantages and disadvantages of bioluminescent probes. And then third, we'll talk about reversing cause and effect and using optics to control biology rather than to read it. So we'll start with the first. So electricity, in some ways, is the fastest biological signal, and for a long time, it's been a difficult signal to make a fluorescent reporter for. There's some very interesting history of electricity. Galvani discovered that electricity was the basis of nerve and muscle conduction using his favorite system, the amputated frog leg. So he would just put the frog leg on a wire, and during thunderstorms, you would see the legs twitch. And it was Dubois-Reymond here in France who first built a galvanometer and used it to show there are electrical impulses as you twitch your muscles, as you contract your muscles. These days, of course, classically we measure voltage using electrodes, and it's measured from the inside out. So a negative voltage at rest means that there are more negative ions inside the cell than outside. Now, what makes voltage interesting is that it's a basis for, of course, our thoughts and actions. And in the brain, neurons act as analog electrical calculators. When the neuron receives neurotransmitters, in the dendrite, there's a postsynaptic potential, which can be either inhibitory or excitatory, so either an IPSP or an EPSP. And then this potential propagates down the dendrite toward the cell body in a mixture of passive and active means. And if the voltage sensed at the cell body reaches a certain threshold, then the action potential is generated. So, and that's an output, which that of course leads to synaptic vesicle release at a distance. Now, the postsynaptic potentials or inputs, as we mentioned, they're low amplitude and they decay over distance. The APs originate at the cell body, at the axon hillock, and they're fast and they regenerate. So these are very different kinds of signals. This is low, and then this is large but very, very fast. The time constant of the action potential is about 1 millisecond up and 1 millisecond down. So voltage imaging goes back a long ways. Actually, when Roger himself was a student at Cambridge, he wrote that he rationally decided in 1972 tried to design and synthesize new dyes for the specific purpose of imaging neuronal activity ideally we would stain the neurons with a dye that would light up or change color whenever and wherever a neuron fired an action potential. He was actually scooped the very next year by Larry Cohen at Yale, who found that the dye myrcene, a fluorescent dye, would demonstrate a fluorescence change upon voltage change. This is in the squid giant axon. So here you can see that the change is actually very small, but the kinetics are very good. But as a chemical dye, there are some major limitations. One is that, well, this change is small, but also neurites and multiple neurons are intertwined, so they're not separable. If you just put a dye on a preparation, you would never tell apart individual neurons. And then the dyes cannot be limited to specific neuronal types. So genetic encoding has the potential to solve those problems. And so we know that through lots of experience now with calcium indicators, and they really show the utility of genetic encoding. So this is a famous GCAMP that a lot of neuroscientists and also many non-neuroscientists use as a calcium sensor. It's actually based on this concept from Roger's lab of two domains that will interact in an analyte-dependent manner, fused to the new N and C termini of a circularly permuted GFP. So you can see that the actual crystal structure looks a lot like the proposal. And the nice thing about genetic encoding is you can target specific cell types, and the expression lasts for months. And then you have some sparsity, so you could tell cells apart from each other. So this is a movie of GCAM in real time in the cortex of a mouse. But one thing you notice about this movie is that it's kind of slow. So most movies of calcium that you see played are played 5 times or 10 times sped up because it's sort of boring watching this. But you can tell immediately that this is a problem because if you're, people and thinking is very fast. This boy is playing Tetris. He played Tetris until the end, until the game broke. So he set the record for longest Tetris game. And in a few milliseconds, he would have to recognize the object, make a decision on where it goes, and then execute the motor commands to turn the object. So that would obviously not be compatible with the speed of calcium imaging that you just saw. All right, so, but calcium imaging is started in 1997 with Chameleon from Roger's lab, but relatively soon, I guess 12 years later, many groups are using GCAM3 because in vivo is able to give a change in fluorescence of about 2.5% to 5%. At the same time, actually, in 1997, the first voltage indicator based on GFP was described by Udi Isakov. But 12 years later, they were still not being used. And there was a lot of pessimism in the field. In fact, there was a review paper that came out when I was a postdoc, and it caused Roger to stop working on this problem. Because the review paper said it was impossible to make a voltage indicator based on GFP and a voltage sensing domain. And some of the things that the paper pointed out that are true is that very few genetically encoded voltage indicators, or GEVIs, can be expressed per cell body compared to calcium indicators. You have to use 50-fold faster imaging to catch the speed of action potentials if your voltage indicator is as fast as the action potential itself. And then GFP has a finite brightness per molecule, as we all know, and its signals must compete with autofluorescent noise. And so that's why there was this prediction that a GFP voltage sensing domain GEVI is unlikely to work, even if it had a response of 10% per action potential. Well, we tried anyway, and we decided to go back to the original concept of a circular permuted GFP and finding a place to put it so that its shape would be modulated by molecular shape changes during voltage changes. So this is a molecular dynamic simulation of a voltage sensing domain, and we see that there's predicted to be this conformational change between the 3rd and 4th helices of the voltage sensing domain, which is actually on the outside of the cell. So Francois Saint-Pierre, who was a postdoc in the lab at the time, placed the CPGFP in the extracellular loop, and indeed we saw a change in fluorescence with membrane depolarization. So that was good. And you can see that the expression of ASAP is at the membrane very efficiently where it needs to be. And the kinetics of the sensor are very fast. So at room temperature, the taus for on and off rates are 2 milliseconds. And at body temperature, they're sub-milliseconds. So they can keep up with APs. And you can see that here, this is a hex cell, non-neuronal cell that's been patched. And we're putting it through different voltage steps. And you can see it's blinking as we change the voltage. So cutting short a very long process of improvement. So over the last 10 years, we've improved it up to ASAP 5. And that, which just came out last year, has a very steep dependence on voltage. So about a 50% change over the 100 millivolts physiological range. And it's as fast as ASAP1 as well. So here's an example where we actually put an electrode, a patch electrode on a neuron, and we can record spontaneous activity. You can see the electrical trace here in the black. So some of these smaller bumps are about 1 millivolts, and you can see that every bump here can be seen here. And also we could keep up with the speed of action potential generation. And this already shows the advantages of imaging, because there's another neuron in the field of view, which we didn't patch. But you could see using voltage that you don't need to patch it. You can see every change as well. And sometimes there are shared inputs and sometimes there are non-shared inputs, just as you would expect. All right, so this, I'll just give you one example of how you can use voltage, how people want to use voltage indicators. The one way of using voltage indicators is to visualize dendritic computation, this process of how signals at the synapses are transduced down the cell body and whether there's decay or integration, et cetera. So because we could see the entire cell at once, it's like having a patch electrode at every place in the cell. And that means that when there's a spontaneous synaptic vesicle release event, we could see it as a local change in the voltage or a local change in the fluorescence. So here we see that looking at this particular site, we see a bump that's happened spontaneously. And then at the soma, with a slight delay, we could see that the bump is there. So that means the voltage is propagated down the soma, but it's much decayed. So here the fluorescence is 15%, here it's 1%. In fact, we could look at various points along the dendrite and we see that the decay follows an exponential curve, which is as predicted from cable theory. There's no regeneration of the signal. In fact, you can put this in a movie and you can watch it in real time. So you'll see a response start here and a decay down to the cell body. Now this allows us to ask a few simple questions about how neurons work. So one question is, how well are distal synapses heard? Naively, well, there's a problem for neurons that synapses are located at different distances from the cell body. If their responses all decay exponentially, then the further located synapses would either see more decay of their contribution, or they would need to have a higher initial voltage so that they can compensate for the distance. And so this concept is called synaptic scaling. And synaptic scaling is absolutely required if you believe in a democratic model of synapses. Although these days we now realize that there's more localized computation and regeneration as well. But this concept, this phenomenon of synaptic scaling has been observed in vivo with distal patch clamping, but just at one location, at one neuron. And we asked whether synaptic scaling depends on tissue architecture, like is it inherent to neurons, or do you need the architecture of the brain? And is it enough to make up for distance? So we could take these kinds of measurements I just showed you and do this for different events that originate at different points along the dendrites. And we could see that the further away the initiation event is from the cell body, the larger the initial fluorescence change, so the larger the voltage change. And this is true for multiple neurons. We just showed two here, one in green, one in red. But interestingly, by the time you measure the voltage change at the soma, you see that there's an inverse relationship. So the further away the voltage had arisen, the smaller it has become by the time it reaches the soma. So that means that indeed there is synaptic scaling in culture. It does not depend on tissue architecture, but it's not sufficient to make up for the distance entirely. So it means that distal synapses they shout louder, but they're still heard less well. All right, so now the primary motivation for doing voltage imaging is to understand the brain. So that, of course, requires looking in vivo. And in vivo, most neuroscientists prefer two-photon microscopy because of its its inherent optical sectioning capabilities. And so I think you all know that in one photon light, you create a cone of light focused to a diffraction limited spot, a point spread function at the focal plane. But the problem, of course, if you're in a tissue that has signal that has fluorophore above and below, you're going to create all this excitation of your fluorophores, which are considered background because then they just add to a diffuse background to the image at the focal plane. And the deeper you go, the more the background competes with your signal. So you're limited to about 100 microns in detection with one photon microscopy in the brain, or about 200 microns. But with two photon, you could go deeper because you're not exciting anything, right? Only at the focal point do you have enough intensity to get the two photon excitation of your fluorophore and create fluorescence. And that's nice then because you can capture all, you don't need a pinhole, you capture all the light that comes back. But the problem with two photon microscopy is that it's inherently a serial approach. You have to scan your excitation point across the sample, and that can be very slow. So to illustrate how slow that can be, so if you have a fast indicator like voltage, typically in a two photon scanning system, let's say you might be scanning at 30 frames per second, in which case you're scanning at each spot, you're returning to each spot every 33 milliseconds. But a voltage signal will only last for a few milliseconds, and so your probability that your pulse actually coincides with your voltage signal is very low. This is not a problem with calcium indicators because calcium inherently has a slow off rate. So once calcium enters the cell after an action potential, it takes several hundred milliseconds to decay. So you can leisurely scan every 33 milliseconds and you'll still catch the presence of the calcium signal. So this means that if you want to scan in one photon, you have to actually scan with a repeat interval of less than maybe 5 milliseconds. Ideally, 2 milliseconds or 1. So how many neurons can we image if you're limited, let's say, with a resonant galvanometer 2 photon, you're limited to about 16 lines every millisecond. Well, so you could decide to scan only 16 lines, so you could scan 16 by 800 spots. That will pack about 30 neurons into that volume. I'll walk us through 3 different methods. So that's like a standard resonant galvo method. You could use FASED, this method that uses mirrors to create a line of dots, which you then scan in one direction. But this limits there's a limit on the irradiance, and thus the single unit SNR. And then you could scan selectively, so you could choose just one spot in multiple locations. You could do this by random access microscopy, multi-photon microscopy, or holography. And this is limited just by how many spots you could do, either by the speed of your method or by the power. So the first method is the most simple one. You just use resonant galvanometry, two photon scanning. In this case, our collaborator Lisa Giacomo in the hippocampus had densely labeled basically all the pyramidal excitatory neurons in the hippocampus. So you could line about 30 neurons up in this very small 16 by 800 pixel volume. But that's enough to allow us to see that there are place cells that are forming in this region. So the famous hippocampal place cells are cells that become active as a mouse explores a new space. So anytime we walk into a new location, cells pick up activity as we're at different parts of that location. And it's thought, of course, to be important for navigation as well as learning places. And so you can see that different cells will be activated at different positions along a track. And you can see this in both voltage with ASAP and calcium with JR-Gecko, a calcium indicator. But if you look closely, you can see that the ASAP peaks are much sharper and more transient than the calcium peaks, as we expect from their different kinetics. The second method is called FASED. And this is work done by our collaborator Nazi at UC Berkeley. So in FASED, you use a cylindrical lens to create a line of lights, which then you send into these slightly non-parallel mirrors. And that means that the light will go to the end and come back. But the amount of distance that the light needs to go is slightly different depending on its initial position along the line. And so that means that dots come out, instead of having a line, dots come out at slightly different times. And based on the length and the angle of the mirror, you could adjust the temporal spacing between the dots. And so Na's lab adjusted the temporal spacing to be about 2 nanoseconds. So similar to the lifetime of the fluorescent protein itself. And so using that, they could put 80 dots along this line and then scan that line of dots in the Y direction. So you basically get a super fast raster scan. The raster scan time is limited only by the fluorescence lifetime that you set, or the interval that you set. So in this case, there are about 30 neurons with ASAP3. 7 of them are shown here. You can see that the neurons share a lot of the subthreshold voltage signal, but the spiking activity is a rare event. All right, and so the last way to perform fast photon scanning is using targeted illumination. And you can do this with random access. We're using acousto-optical devices. So this is work that's been pioneered by several labs around the world, but most notably by our collaborator Stéphane Dieudonné and Laurent Bourdieu at École Normale Supérieure in Paris. And so this movie made by Kurt Haas shows that as you put in different frequencies of sound through these AODs, the angle of the, let me just play that again, the angle of the light coming through will change. And that allows you to steer the laser beam to different points. Right, and so using this, they could image 3 neurons expressing ASAP3 deep in layer 5. So this is 500 microns below the surface, or 440 microns below the surface. So these are just 3 examples of voltage traces that can be applied looking at spikes and up and down states. More recently, we used this very expensive Femtonics Femto3D Atlas from Hungary. And this has the AOD in the Z direction as well. So this allowed us to image about 10 neurons that span a Z space of almost 300 microns. And you can see the nice voltage traces along different positions. All right, so that's how you could do fast 2-photon scanning. But actually, you know, the convenience of 1-photon scanning is still much, much easier. And actually, 1 photon is sufficient if the sensor is high contrast. And we know this very well from experience with GCamps. So in this example, you get to see some place cells in action. So this is actually a 1-photon image of the hippocampus as the mouse is running. You can see that here, there's a cell that will light up when the mouse passes this triangular object on the way down. So right now it lights up. It's interesting, it's both a place cell because it marks this location, but a direction cell because it only occurs when the mouse is going down. But this is all 1 photon, so you can see it's quite easy. So the condition to use 1-photon microscopy is that your probe has to be high contrast because it needs to have very low, it has to be a dim at rest and bright upon induction because you want the background to be low. Right? And so that requires flipping the response of ASAP. So ASAPs had been bright at rest and dark upon activation. So we had to flip the response. So we did that recently with ASAP4. So it responds about 200% in the positive direction. And we've recently, in unpublished work, we've made an ASAP6. And you can see in this trace the difference between GCamp and ASAP6 due to kinetics. Interneurons fire many action potentials all the time. And in GCamp6, you sort of get this averaging effect where a lot of calcium builds up in response to a burst of action potentials, but you can't really discern each individual action potential. But with ASAP6, you can see each AP in a near binary fashion. And this is a movie being played in real time. And actually, you don't see most of these because they're too fast, right? They're only there for 2 milliseconds. That's faster than a blink of an eye, so you don't actually see individual APs. When you do see flashes, you're probably also seeing bursts of action potentials. But you can see that even the bursts are very fast. All right, so one interesting thing about 1-photon is that you could turn the cone of light into an asset. because you're not only exciting at the plane of focus, you're exciting everywhere. And so we did this very simple thing where we just put we just split the light path and put a slightly defocusing lens in one light path. And that allows us to image 2 planes at once onto two halves of a CMOS chip, right? So very, very simple. This allows us to double the number of neurons we could acquire in a single frame. You're going to take a hit on the SNR because you're losing half the light on each frame, right? But that hit is on the order of square root of 2, right? The signal to noise ratio is dependent on the square root of the intensity of light. But as a result, we can image 200 neurons at once. So this is now, I think it's the first time. Previously, the record was about 40 neurons, but now we can increase that by a factor of 5. And you can see actually in the hippocampus that there are different kinds of action potentials. There's a single action potential, and there are these so-called complex spikes. And the complex spikes are believed to form when the neuron first forms place cells. So when place cells first arise, they are, oh, thank you. Merci beaucoup. Okay, can you hear me? Is it working? Yes, okay, thank you. So when place cells first arise, a complex spike is formed. So this is a movie, again, in real time, and you can see very fast spiking everywhere. And again, you don't actually see individual spikes. You probably will see the complex spikes because they last a little bit longer. And actually, a very interesting thing is this work is using a global shutter CMOS from a French company called First Light near Aix-en-Provence, Méheur. And it's, as far as I know, we're the only biologists to use this camera. It was developed for astronomers, but it's not even that expensive compared to other CMOS cameras. It's about 22,000 US dollars, maybe 20,000 euro, and it can image about 1,000 by 1,000 pixels at 800 frames per second, and it's true global shutter. So it's great. All right, so I think now we can get closer to thinking about, to discerning how neurons operate, when in real time at the speed of thought. So, this sort of Tetris game, you can imagine trying to deconvolve its individual actions from the actual voltage signal. Right, so in summary, voltage imaging is possible with GFP and voltage sensing domain, thankfully. The SNR improvement is still desirable for in vivo experiments. It's currently still limited by the short exposures required, the low fluorophore numbers, and then the maximum tolerated power. So you might wonder, well, if you want better SNR, why not just boost your power? We're already operating at sort of thermal limits for the mouse brain, which are about 100 milliwatts per millimeter squared. So, and then, these kinds of things like the short exposures, the fluorophore numbers, and the power, they have square root dependence on the SNR. So there's some limits, it's not a linear gain as you increase fluorophore number, for example. But improving brightness or responsivity will improve SNR. So by the square root on the brightness and by linear on the responsivity. So making it more responsive is probably the most important thing. And then finally, if you can improve the illumination targeting efficiency or the emission collection, this will also improve SNR. But again, this is by the square root of how much you improve. All right, and so as I mentioned, with the synaptic scaling experiment, we can now use GEVIs to reveal unexpected features of electrical signaling neurons. For example, there's other groups using GEVIs for visualizing synaptic weight changes upon sensory or optogenetic stimulation in vivo. All right, so that's the first part. Let's see, how am I doing on time? It's been 29 minutes. Okay, last 2 parts are shorter. So now we're going to go to something totally different, which is bioluminescence. And I call this photon watching because bioluminescence, you don't excite, right? So it's not an active kind of imaging. It's a very passive kind of imaging. And so there are advantages and disadvantages to that. So, when fluorescence first came out, there was this beautiful cover image from Marty Chaffee on looking at GFE expression in neurons of the worm. And it's this beautiful high contrast, high resolution image. Around the same time, Chris Contact at Stanford used firefly luciferase to image the presence of bacteria in a mouse. And it's grainy, you know, and they always use this pseudo color because I guess the original image doesn't look so great. And so I've always wondered, why is it that luminescence imaging is used and why is it so poor in quality? So Roger in 2003 wrote this review on different kinds of imaging modalities. So there's a nice chart here of all the different ranges that different modalities use. And our favorite fluorescence, he wrote, can be used down to 1 millisecond and in about 0.3 microns. Of course, now with super resolution, we can go to 0.1 microns and we can go faster in a millisecond, but it's close enough. And then he wrote that bioluminescence is sort of confined to this upper right corner of low spatial resolution and low temporal resolution. Nevertheless, even despite this so-called 10-second limit, you need long integrations. Nevertheless, people use bioluminescence because you can have lower limits of detection due to the absence of background. So there's no autoluminescent background. So you can just integrate you can take 10 seconds to integrate your photons and you can get a signal. But we were curious, can we improve the speed and resolution of bioluminescence imaging? Can we push it to be more like fluorescence? And can we extend bioluminescence to molecular reporting just as people do with fluorescent proteins? I mean, photons are photons, so if you can get more photons out, you should be able to do both. All right, so our favorite marine biologist, Osamu Shimamura, also, he was actually more interested in luciferases. Apparently, if you asked him about GFP, he would be annoyed because he really cared about aquarin, which is the fret donor to GFP in jellyfish. And aquarin is a calcium-activated luminescent protein. But it has basically no catalytic activity. It has a zero turnover enzyme. But he also found a luciferase from the deep sea shrimp, Oplophorus, which then Promega engineered into a very efficient monomer called Nanolook. And probably some of us have used Nanolook. It's very popular in vitro. How many of you have used Nanolook? Yes, many, excellent. Yeah, and so Nanolook has about 100-fold higher photon production rate than firefly luciferase. So we got into this because Jun Chu in the lab when he was a postdoc developed a protein called Psi-OFP, cyan excitable OFP, which is really nice, juicy color. And we noticed that because it's cyan excitable, it's a good fluorescence, Forster resonance energy transfer receptor or RET receptor to Nanolook. And it's so that means its excitation spectra overlaps with the emission spectra of Nanolook, and we made this protein Antares that is a fusion of 2 copies of Psi-OFP with Nanolook, and it has this nice RET. Some people call it BRETs for bioluminescent resonance energy transfer, but I think in Europe it's more, it's recognized that BRET is Forster resonance energy transfer just from a bioluminescent donor, so I just call it RET to avoid any confusion. All right. And the nice thing about Antares is that by shifting the emission to 600 to a considerable amount over 600, you can bypass hemoglobin absorbance and get better transmission through mammalian tissue. So we show this by using Antares in the liver and then performing bioluminescence. This allowed us to test many different substrates to improve in vivo substrate usage for Nanolook. So working with Promega, we developed furimazine, and then later, we developed fluorofurimazine, which you can see has about 3 to 4 times the brightness of the original furimazine. Oh, that's hydrofurimazine. Yeah, what am I missing here? Something little light green. Oh, okay, yeah. So anyway, we have fluorofurimazine, which is better than this intermediate hydrofurimazine. And we can get very nice images in the liver without any surgery, right? So there's no dissection. This is a living mouse, but you could see through the skin and into the liver. And then later on, we made cephalofurimazine for use in the brain, so you can get these nice images of the brain, which are about 100 times brighter than with firefly luciferase. Again, you could get very nice anatomical images without dissection. And it's bright enough that you can get real-time voltage rate imaging. So this is acquiring at 30 frames per second. with anteriors expressed in the brain, so you can track mice in real time. All right, so I was saying that it'd be interesting if we can make bioluminescence report biochemical events just like we do with GFP routinely. And so we first started with everyone's favorite events, calcium. And just like with GCAMP, we can fuse calmodulin and M13, which create a complex in the presence of calcium. By fusing it to nanolook, we can have a situation where the nanolook catalytic activity is low, and with calcium, it becomes higher. So in this case, about 7 times higher. And we can get different affinities by changing the linker length, just as we do with calcium indicators. So now we can start using it in a sort of, even in microscopy if you wanted to. There's really, most times there's no reason to do this, but it occurred to me that bioluminescence can act as a no excitation microscopy channel. So if you're really limited for channels, you could put a bioluminescent indicator in there, and it would be the channel where you don't excite, right? So there would be no fluorescence, but you can pick up bioluminescence. So here are 2 examples. These are HeLa cells undergoing calcium oscillations when treated with histamine. And then this is a cardiomyocyte differentiated from an ES cell that's doing spontaneous beating in vitro. All right, but the real reason to use bioluminescence is to perform sort of this macroscopy, sort of this large level imaging where you don't get cellular resolution, but you can target specific cell types in vivo. So here's an example of calcium imaging in a brain, again, without any surgery. So we're just putting a mouse in a black box and then using a camera. I mean, the amount of luminescence is large enough that if you just, let's say, put nanolook in the liver, anteriors in the liver, and inject a substrate and go into a dark closet, you could see it with your eye. So it's plenty bright. So another signal that we're interested in is kinase activity, which we've seen talks about here from Michelle and others. So kinases are major drug targets in the brain, so we decided to develop methods to visualize drug activity in the brain. So we made 3 sensors, one for PKA, one for ERK, and one for AKT. So these are 3 different kinases. And the sensor designs are generally like this, where we split nanolook into large bit and small bit. These are two complementary fragments of nanolook developed by Promega. And small bit is simply the last beta strand on the barrel. And we peel away small bit using a phosphodependent interaction. So a phosphorylated substrate would bind to a phosphopeptide binding domain. And we use that to peel away small bit from large bit. And the idea is then when an inhibitor of the kinase comes along, let's say this is ERK, when an ERK inhibitor comes along, then eventually phosphatases take over the situation, and then the equilibrium shifts to a dephosphorylated form, which is allowed to complement back and be luminescent. So this is what the models show it to look like. You can have a small phosphopeptide binding domain, like the WW domain. We can have a large one, like the 1433 domain, but it works in either case. So here's an example where we put in the ERK. These are called KIMBs for kinase modulated bioluminescent indicators. We put the ERK KIMB into the brain by using AAV, and then we could test 2 different ERK inhibitors. One is called actually these are MEK inhibitors, which is upstream of ERK. So one is mirdametinib, another is trametinib. And mirdametinib is known not to cross into the brain, and trametinib is known to cross into the brain. Actually, sorry, it's the other way around. So you can see that after mirdametinib, we get more luminescence. After trametinib, we don't. And this is statistically significant. But we always do a dead KIMB. We always have a KIMB that doesn't get phosphorylated as a control against any sort of or blood flow effects of the drug itself, because blood flow influences the delivery of the substrate. What's nice about this is that you can do so-called pharmacodynamics. So in drug discovery, pharmacodynamics means you have to inject drug, you have to kill the mouse, you take out an organ and you look at the target. So let's say it's ERK, you might look at phospho-ERK using Western blotting. So that's obviously very time consuming, but also uses a lot of mice. And it is also highly variable because every injection, every dissection is different. But with KIMB, you can express KIMB in a single mouse. You can inject a drug once and you just perform bioluminescence imaging at different times and you get a nice curve that shows how long the drug stays active in the brain. And when this is over, you can use the same mouse the next day for a different dose of drug or a different drug. And so you can see here that we have 2 mice. This is with intracranial tumors expressing KIMB. This is with AAV expressed KIMB in the brain. We get the same time course, but there's a different amplitude. And that shows that the tumor situation is more blood-brain barrier penetrant. Well, the blood-tumor barrier is disrupted, so there's more exposure of the tumor to the blood. All right, so we could do the same thing with AKT KIMB. And what's interesting with AKT KIMB is that it reports that pharmacodynamics exceeds pharmacokinetics. So if we look at the plasma concentration of a drug, capivasertib, we see that it decays very fast. The half-life is only about 2 hours. But the KIMB response actually doesn't peak until 4 hours later, and then the half-life is something like 12 hours. It's incredible. This is true for across AKT drugs, so it's true for this other drug, ipatasertib, as well. This is what it looks like in real life. So we always put on these raw luminescence images because I think now it's good enough. It's like fluorescence. You don't have to hide behind these sort of pseudocolor images. But we have to put a pseudocolor on because that's what people are used to in bioluminescence. It's one of these protacs, which are these degraders, molecules that cause degradation. After a single injection, we see that the protax half-life is something like 4 days. It's incredibly long. Yeah, so you can see interesting, unexpected pharmacodynamic effects this way. All right, so I think then that we have managed to move bioluminescence a little bit more like fluorescence. It's still not as fast because, again, you're talking about 100 photons per second versus, versus 10 to the 9 photons per second. So it's a huge difference. But it's good enough that you can see it under a microscope. And the brightness can be modulated by calcium and kinases, similarly to GFP. And we can use this for excitation-free microscopy or non-invasive macroscopy. All right, so let's see how much time do I have. Okay, it's been 42 minutes, good. So we'll go to the last part, which is reversing cause and effect in photoproteins, or using photoproteins for optical control of biology. So everything we talked about right now is, with reporters, is biology influencing the production of light, right? But can we reverse this effect? Can we use light to control biology? Because imagine we could do that, then we could really investigate signal transduction, cause and effect in biology. Now, this is not a new idea. Many people have been interested in this. In fact, plants do this all the time, and so there's been a lot of motivation to take proteins from plants that respond to light and use them in mammalian systems. But opsins themselves in our eyes are an example of fast biological responses to light. And of course, there are opsins in bacteria, and that's where the famous channel rhodopsins come from. So one kind of question that would be very useful to investigate with optical control are complex morphological events, right? And so an example of that is this neutrophil that's chasing a bacterium in this famous movie. You can see that the neutrophil senses where the bacterium is, and can respond. And so something in the cytoskeleton of the neutrophil is very dynamic. And through a lot of work in the field of cell biology, of course, we know that Rho family GDPases are important in controlling different aspects of morphology. So Rho A usually promotes contractility or adhesion, and then RAC and CDC42 promote the formation of lamellipodia and filopodia. So you might imagine that directional control of these 3 Rho GDPases would be important for directing migration. This actually interest of mine that goes back to my PhD work with Mike Greenberg, where we were studying axon guidance using F receptors. So when you activate an F receptor, in this case in this hippocampal neuron, you see that there's this immediate retraction and loss of adhesion. Now, other people have implicated Rho A in this process, which makes sense because Rho A is usually contractile. And in fact, we made this Rho A right shoe that Michelle also mentioned, which showed indeed that Rho A is activated at the growth cone very soon after F receptor stimulation. And there's this actually long-lived, much delayed activation in the cell body. If you do a Western blot, you see the kinetics of Rho A peak at 20 minutes because you fail to detect a very small amount of Rho A. So this shows the advantage of using using FRET reporters for local investigations. But RhoA, I mean, this observation raised a conundrum because RhoA is better known in the cell biology field for promoting adhesion, whereas we see the loss of adhesion of growth cones. And this RhoA activity in cell adhesion is usually done by transfecting or injecting a constitutively active RhoA, and you get the formation of stress fibers and focal adhesions. So we were curious if we can perform spatial temporal control, sensing of protein activity using FRET reporters. Could we also have a general method for spatial temporal control of protein activity using light? So we were thinking a long time about how to do this efficiently. And then we realized that the answer may be GFP again. So a green fluorescent protein DROMPA was characterized by Tsushi Miyawaki to undergo a loss of structure along one side of the beta barrel in response to light. And this corresponds with a cis-trans isomerization of the chromophore. What's interesting is this, in the ground state, the protein absorbs at 500 nanometers like other GFPs. When it's photo-switched, it then absorbs at 400 nanometers. So you could go back and forth with DROMPA. And I think many of us here have used DROMPA as a probe for super resolution microscopy. So Shinjo, when she was a student in the lab, she engineered DROMPA into a dimer, which then will be photo-dissociated with 500 nanometer light. So the idea is that loss of structure actually exists along a native dimeric interface. So by restoring that interface to just the right strength, we're able to cause that loss of structure to occur with photoisomerization and lead to a loss of dimerization. And then we could switch this back with violet light, so it's bidirectional. So we can see that here, it's dimer at baseline. We put on 500 nanometer light, it becomes a monomer. And when we switch it back with 400 nanometer light, it becomes a dimer again. What's nice about this system is not only is it bidirectional, but it's completely genetically encoded, of course, right? You don't need any cofactors. And it self-reports, so it tells you when it's switched off because it becomes dark. And so using this dimeric photodissociable dimeric DROMPA or PD DROMPA, we're able to control a variety of proteins of interest. So the idea is that if we can fuse 2 copies of PD DROMPA to a protein of interest at the right locations, the dimerization at baseline could occlude an active site, and then we could photo switch it on with cyan light and photo switch it off with violet light. So an example are kinases. We could fuse a DROMPA to the N-terminal lobe of a kinase and another one to the C-terminal lobe, inserting into a loop. And then the dimer will form in front of the active site and block access to the substrate. And so we show in the case of MEK, which is a kinase upstream of ERK, that we can control the phosphorylation of ERK. We could basically recapitulate a similar dynamic range as CRM stimulation to starved cells, right? So it's pretty efficient. And this is what a model of it might look like. This is not a real structure, but just a model. So you can see that the 2 DROMPA dimers are very large, including the active site, but are opened up upon photo illumination. Right, so getting back to Rho GDPases, Zia Sung, when she was a postdoc in the lab, made a photo switchable Rho GEF, PS Rho GEF. And so this is an activator of Rho GDPases. We fuse it to a membrane signal, so it's residing at the membrane. When we activate it with light, it should activate Rho A. We can see that here, so if we look at the amount of Rho A GTP, it goes up in proportion to the amount of lights that we put onto the system. So now we could finally ask, is Rho A sufficient to induce growth cone retraction? So here there's a growth cone that's extending, and when we hit it with light, we see this very rapid retraction as we'd expect. So Rho A is sufficient for cause de-adhesion. So now we could ask, well, what's the difference between neurons and cells, non-neuronal cells? Why is it that people say Rho A activates adhesion? So we put Rho A in a non-neuronal cell. These are U87 cancer cells. We see that actually we can get retraction just fine. In fact, we get the loss of focal adhesion. So there's a huge group of focal adhesions that dissolves away. Right? We see this repeatedly. So eventually we worked out that the difference is in the amount of Rho A activity. So you can control Rho A rheostatically with PS Rho GEF. And we see that as we put on more light, we get more loss of focal adhesions. But at a certain point, we actually stop having that effect. We get no loss of focal adhesions. And in fact, if you look carefully, you can see some focal adhesion growth. So you can see that here, as we put on, when there's no light, there's no change. As you put on an intermediate amount of light, there's a loss of focal adhesions. If you put on more light after that, you get no loss of focal adhesions, in some case, a growth of focal adhesions. We always do a drumpa only control to make sure we're not having effects of light itself. And so this is a case where optical control allows you to decode not just the time of a response, but also the intensity of a response. All right, and so there's a lot more work with optical activation of Rho done by Mathieu Copy at Institut Curie. And so he wrote this nice review on optical approaches to study Rho. So, you know, I talk about drumpa because it's our favorite, but there are many other ways to control proteins with light. They usually are to induce relocalization of proteins through a heterodimerization effect, such as Cry2-Sib, or a love domain with a binder, or this love domain that spontaneously goes to the membrane with light. So I would say, actually, I would admit that these are more popular because they're easy to use. You just have to relocalize proteins. You don't have to create a new construct. But we believe that creating a new construct is useful because you don't overexpress active proteins. You create a cage protein. All right, so in summary, the drumpa GFP can be engineered to a photodissociable reassociable dimer, making it a bidirectional photoeffector. PD drumpa self-reports and does not require any cofactors. And then it enables the construction of single-chain photosocial proteins. And an example of that is photosocial ROGEF revealing rheostatic control of cell adhesion by Rho. All right, so that's the whole talk. I've told you about fast sensing with GFP, slow sensing, photon watching with bioluminescence, and then reversing cause and effect with optical control. And so finally, I just thank you for listening and thank all our collaborators, the people in the lab, and all the collaborators we mentioned along the way, but especially Stéphane Dudonné and Laurent Bourdieu at ANS. Thank you very much.
Michael, are the Kimby sensors reversible? Like, will large bit and small bit dissociate upon dephosphorylation?
Yeah, it's a great question. Yeah, large bit and small bit themselves are inherently very, very low affinity. The restoration kinetics are not dependent on the on Kimby on small bit, large bit, but they are dependent on the rate of dephosphorylation in the case of the kinase indicators, or rephosphorylation, right? So we put on. Okay, so the onset requires dephosphorylation. The way we design a kinase indicator, right, they're kinase inhibitor indicators, right? So they become more luminescent with inhibition of the kinase. So that requires the phosphatase to go in and remove the phosphate. So the on rate actually of the Kimbys is limited by the strength of the phosphopeptide binding interaction with the phosphopeptide. So we've noticed that it takes about 10 minutes, and so we're not sure if the drug is active. Let's say the drug is active at 0 minutes and we had to wait 10 more minutes for the dephosphorylation event, or whether the drug itself takes 10 minutes to inhibit. So there's a loss of time resolution there. Yeah, but it's for pharmacokinetics, we think that resolution is okay.
Yes. Thank you for the. Hello. Okay, maybe I can ask.
Sure. Yeah, it's a great question. So the question was, how much energy is stored in the chemical, and is it going to disturb any biochemical processes in the cell? Yeah, I mean the luciferase substrates are substrates that are oxidized, so they require oxidation by O2. They do undergo spontaneous release, but very, very long over time, like hours. Yeah, so probably it's by that time the substrate is peed out of the system or oxidized in the liver. It's not clear what the clearance mechanism of the luciferases are. But yeah, I mean, they're not inherently that reactive on their own. So that's why, you know, you need a luciferase to react them.